Hello and welcome to this session. LO4, demonstrate an understanding of medical applications within modern genetic engineering techniques. Today we will explore how modern genetic engineering is transforming medicine. By enabling precise modification of DNA, these techniques play a vital role in advancing diagnostics, developing targeted therapies, and supporting personalized medicine. Recombinant DNA technology involves inserting a gene into a host organism to produce a protein. The process includes identifying and isolating the gene, cutting DNA, and inserting it into a plasmid vector. The plasmid is transferred into a host cell, like bacteria. The host cells express the gene to produce the protein. This technology is crucial for producing therapeutic proteins. Human insulin is produced using genetically modified bacteria, reducing allergic reactions compared to animal-derived insulin. Human growth hormone treats growth deficiencies, while clotting factors aid hemophilia treatment. Monoclonal antibodies are used in cancer therapy and autoimmune diseases. Recombinant DNA allows large-scale production of therapeutic proteins. This technology ensures purity and safety in medical treatments. Gene therapy modifies or replaces faulty genes to treat diseases. Somatic gene therapy targets body cells, while germline therapy targets reproductive cells. Viral vectors deliver functional genes, and non-viral methods like lipid nanoparticles are used. It treats inherited disorders, certain cancers, and retinal disorders. Gene therapy addresses root genetic causes rather than symptoms. CRISPR. Cas9 is a precise tool for targeted DNA modification. Guide RNA directs the Cas9 enzyme to a DNA sequence, cutting it for repair. This allows gene insertion, deletion, or correction. It's used for correcting genetic mutations and researching cancer genetics. CRISPR offers high precision and efficiency in gene editing. PCR amplifies small DNA amounts, enabling rapid and accurate diagnosis. It's used to detect infectious diseases, identify genetic mutations, and for prenatal screening. The process involves adding ingredients, denaturation, primer binding, and DNA synthesis. Each cycle doubles the DNA amount, producing millions of copies. PCR is essential in medical diagnostics. Personalized medicine tailors treatments based on genetic profiles. This approach enhances treatment efficacy and patient outcomes. Genetic modification of stem cells allows tissue regeneration and blood disorder correction. Modified stem cells are used in treating certain blood cancers. They help develop disease models for drug testing. This technology offers new treatment avenues for various conditions. Stem cell engineering holds promise for regenerative medicine. Genetic engineering raises concerns about off-target effects and long-term safety. Germline modification ethics and accessibility issues are debated. Genetic privacy and cost are significant considerations. Strict regulations guide clinical use to ensure safety. Ethical frameworks are essential in navigating these challenges. Techniques like recombinant DNA, gene therapy, CRISPR, PCR, and sequencing transform medicine. They allow therapeutic protein production and genetic defect correction. Early disease detection and personalized strategies are possible. These advances improve diagnosis, prevention, and treatment. Genetic engineering significantly impacts medical science. Bacterial plasmids play a key role in recombinant DNA technology. They act as vectors to carry genes into host cells. Plasmids are ideal due to independent replication and easy manipulation. They are used in large-scale insulin production for diabetes treatment. This application highlights genetic engineering's medical impact. Insulin gene isolation involves identifying and extracting it from human cells. Messenger RNA is converted to cDNA using reverse transcriptase. DNA polymerase synthesizes the second DNA strand. This produces double-stranded DNA for insertion into plasmids. Enzymes are crucial in preparing genetic material for engineering. Plasmids are small, circular DNA molecules in bacteria, acting as vectors. They carry foreign DNA into host cells and replicate independently. Plasmids are easy to manipulate and can carry marker genes. Viruses can also be vectors, but plasmids are common for insulin production. This highlights their versatility in genetic engineering. Restriction enzymes cut DNA at specific sequences, creating sticky or blunt ends. The plasmid and insulin gene are cut with the same enzyme for compatibility. DNA ligase joins the gene into the plasmid, forming a recombinant plasmid. This contains both bacterial and human DNA. Ligation is essential for gene insertion. Promoter sequences are crucial for gene expression in plasmids. They signal bacterial RNA polymerase to begin transcription. This ensures the inserted gene is actively expressed. Without a promoter, the gene wouldn't be transcribed or translated. Promoters are vital for successful protein production. Transformation introduces recombinant plasmids into bacterial cells. Methods include heat shock and electroporation to increase membrane permeability. Once inside, bacteria replicate and express the insulin gene. This process is crucial for producing recombinant proteins. Transformation is a key step in genetic engineering. Not all bacteria take up recombinant plasmids, so selection is essential. Plasmids often carry antibiotic resistance genes for selection. Bacteria are grown on agar with antibiotics, and only those with plasmids survive. Replica plating confirms which colonies contain the plasmid. This ensures only modified cells are cultured. Selected bacteria are transferred to industrial fermenters for large-scale production. Fermenters provide controlled temperature, optimal pH, and adequate oxygen. Nutrient supply and sterile conditions are maintained. Bacteria multiply rapidly, producing large quantities of insulin protein. Culturing is vital for mass production. Recombinant insulin treats type 1 diabetes and advanced type 2 diabetes. It offers advantages over animal-derived insulin, being identical to human insulin. This reduces allergic reactions and allows consistent production. Recombinant insulin is highly pure and safe for medical use. This application showcases genetic engineering's impact. Plasmid-based recombinant technology produces human growth hormone for growth disorders. Clotting factors for hemophilia and certain vaccines are also produced. Therapeutic proteins are manufactured using this technology. These applications demonstrate the versatility of genetic engineering. Plasmids are central to producing various medical treatments. This is the conclusion of the video. Bacterial plasmids are crucial in recombinant DNA technology, carrying human genes into bacterial cells. Gene isolation, cutting, ligation, transformation, and fermentation enable large-scale protein production. Insulin production exemplifies genetic engineering's impact on disease treatment. This technology improves patient outcomes significantly. Feel free to send a WhatsApp message to 07484361688 if you need assistance. Our team is here to help. Thank you for your attention, and we'll see you next time.